I'm Michelle Dennis, and I'm going to tell you a little bit about our research project that was funded through the One Health Initiative Seed Grant Program, and we'll be investigating freshwater mussel mortality here in the Clinch River, which flows from southwest Virginia through northeast Tennessee. And this next slide shows another view of the Clinch River, which happens to be the most biodiverse river system in North America. This is a treasure of our region. It supports very unique and endangered populations of aquatic species. In terms of mussels, it harbors 46 different species, nearly half of which are endangered. So it's not surprising when these mussels started to die in 2016, it made the headlines. It also got a lot of attention because of the importance that mussels play in the overall health of a river basin ecosystem. They have a critical role in water purification and nutrient cycling as filter feeders. So their loss from an ecosystem can have drastic flow-on effects for other species dependent on that ecosystem. This picture is from another news article that was published a few years later. And sadly, seasonal mortality epidemics have continued since 2016, and there is still so much we do not understand. One of my fellow co-investigators labeled this picture so that you can appreciate the potential scope in terms of the number of species impacted by these mortality events, and also appreciate the large percentage of them which are endangered species. And the causes of these mortality events in the Clinch and those mortality events affecting other mussel populations in other locations of North America remain poorly understood. Our research team came together because we want to help improve this understanding. And so what we plan to do is to place sentinel mussels within silos at multiple impacted sites in the Clinch River, where they will be maintained for a year and periodically assessed. And we'll be looking at them for their growth, survivorship. We'll be looking to see if they have any clinical signs of illness. We will sample hemolymph to look for changes in cells and biochemistry. We will conduct comprehensive postmortem examinations and assess their tissues for any abnormalities, both with the naked eye and at the microscopic level. We will do a microbiome assessment to look for shifts in microbial composition of gut and hemolymph. This longitudinal approach to assessing their health will help us determine abnormalities that are specific to mortality events, and this is going to bring some clarity to generating hypotheses around disease causation. In another arm of this project, the McClung Museum of Natural History and Culture at the University of Tennessee archived several thousand specimens derived from previous years of mortality. And these have been sorted by species and location and date, so it's quite a collection, and their analysis will help us better understand if there's a certain size class or species that are relatively more susceptible to mortality. Finally, we will bring the knowledge that we learned from these two different study arms together and what is already known regarding spatial and temporal trends in mortality, and our team plans to build models to contrast predicted scenarios in mortality for different categories of causative agents using different modes of transmission.